Welcome children. Today we'll continue our discussion on seeds. Now if you remember, on the previous turn we had talked about a dicotyledonous X-albuminous seed, that is bean seed, and we had discussed its structure as well. And today we'll be talking about a monocotyledonous albuminous grain, and that is maize grain. So first question which must have come to your mind is, why do we call it a grain and not a seed? And reason is, this is an LS of maize grain shown here, and you can see the outermost blue layer here, which is a fusion of pericarp and testa. It's a fused product of pericarp. Now what is pericarp? Pericarp is the outermost layer of a fruit, and testa is the outer coating or integument of a seed. So the fruit wall and the seed wall get fused here. So we can neither call it a seed nor we can call it a fruit. So what do we call it instead? A grain. So technically maize is a grain, although it's been explained under the lesson seed. At your level, if you call it a seed also, it's okay. But you need to understand that technically it is a grain, because the pericarp or the fruit wall is fused with the testa or the seed coat. Now you can see the shape of the maize grain. All of you must have eaten corn or maize. This is a single grain from a complete maize cob. This is what it looks like from inside. And unlike bean, which has two cotyledons, maize has only a single cotyledon. So we are now comparing the two. Bean is dicotyledonous and maize is monocotyledonous because it has only a single cotyledon. Bean is a seed because outermost covering is what? Testa. But maize is a grain because outermost covering is what? Pericarp fused with testa. Bean was storing its food in the cotyledons. That makes it X-albuminous. But maize is storing its food in the endosperm, which makes it albuminous. Now in bean seed, we talked about a visible white oval mark which is called the hilum, and close to it a tiny opening called micropyle. But here neither hilum is visible nor micropyle is visible. So these are the main differences if we talk about the two. Bean seed is dicotyledonous, maize grain is monocotyledonous. Bean's outermost covering is testa and maize its outermost covering is pericarp plus testa. Bean is storing its food in the cotyledons and maize is storing its food in the endosperm. In bean, hilum and micropyle are visible, but in maize, as you can see, it is not visible. Micropyle indeed is there. Every seed needs micropyle to take up water, to take up gases, but here it is not visible. Now we'll discuss the internal structure of a maize grain, which has been shown here. This is a LS of maize grain. LS means longitudinal section. Outermost layer is already clear to you. Inner to this layer, we have this green colored layer. I've made it in different colors, not because it's green, just so that you can understand what I'm talking about. This layer is known as allurone layer. This allurone layer is rich in proteins. It's storing food in the form of proteins. Now you can see this entire maize grain is divided into two regions. This upper region is the endosperm, which is storing food for the seed in the form of starch, and lower portion is the cotyledon. Maize has a single cotyledon, and this cotyledon is called what? Scutellum. This is a single shield-shaped cotyledon which is called the scutellum. So this entire area is the endosperm, and what is the function of endosperm? It stores food in the form of starch. So starch is present in endosperm, and in the allurone layer what is present? Proteins. So this is a protein-rich layer and this is starch-rich region. Now in between the two regions, main regions of the maize grain, you can see this dividing layer which is labeled as the epithelial layer. This epithelial layer releases enzymes. When it secretes enzymes, the enzymes are going to dissolve the food present here, the starch, the proteins, and make it available to the scutellum or the growing or developing baby plant which needs the food. So function of epithelial layer is to secrete enzymes and make food available to this developing embryo which is called scutellum. Now, like we studied, what are the parts of an embryo? It has an embryonic axis having plumule and radicle. So here also you will see a plumule, and here you will see the radicle. Plumule which will form the shoot system, and radicle which will form the root system. But here something additional is present which is not present in bean. And what is that? Two sheaths or protective coverings called the coleoptile and the coleorhiza. Another difference now being highlighted between maize and bean. The coleoptile protects the plumule and the coleorhiza protects the radicle. Now I know you find it confusing because lot of new terms are there. So I'll teach you a trick how to remember. Now coleoptile has a P in it, and P is for plumule. So next time you have to choose between the two, let it be very clear. Coleoptile with a P protecting the plumule, and coleorhiza with an R protecting the radicle. So this is how you are going to learn it in case you get confused. So these are two additional coverings present over the plumule and over the radicle only in the grains. So coleoptile and coleorhiza. I hope all parts and their functions are clear to you in a maize grain. So we have pericarp and testa, which is protective. We have alurone, which is storing proteins. We have endosperm storing food in the form of starch. We have epithelial layer releasing enzymes, digesting the food stored here and making it available to the embryo. We have scutellum or a shield-shaped embryo giving rise to a baby plant. The plumule forming the shoot system. The radicle forming the root system. Remember, the radicle here is CLE. The one you study in chemistry is CAL. So this is RADICLE. We have coleoptile protecting what? P stands for plumule. And we have coleorhiza, R for radicle, protecting the radicle. Now we'll talk about a very important step called germination. You all know the seed, when sown in the soil and given the favorable conditions, it germinates. But every seed doesn't start germinating immediately when you sow it in the soil or give it the conditions. It doesn't start germinating. It needs time, and that time period is called dormancy. Now dormant normally means sleeping, but mind it, the seed is not sleeping here. The seed is getting ready for a major change in its life. It has to give rise to a new plant. So this dormancy period is the time period of rest. Apparently the seed is resting, but inside it is undergoing a physiological maturation to prepare itself to germinate or to give rise to a new plant. And every seed undergoes a period of dormancy. Once that dormancy is over, the seed will germinate and give rise to the new plant. But for germinating, after dormancy is over, it requires 3 conditions. What are they? Water, you all know. Air also you know, air or oxygen. But one thing where you get confused is, you often state the third condition as sunlight. It is not sunlight. Every seed doesn't need sunlight for germination. The third condition is what? Suitable temperature. So next time someone asks you the 3 conditions, quickly you should state water, oxygen and suitable temperature, not sunlight for all. And suitable means normally between 25 to 35. We can also call it optimum. Optimum means just right. Now let's see what every condition does to a seed. First, water. First thing which water will do is, it will make the seed coat soft. It will soften the seed coat. Why does the seed coat need to be softened? Because the radicle has to emerge out. It has to break out from it. So first function of water is, it softens the seed coat. And second is, it activates the enzymes. All chemical reactions inside the seed take place due to enzymes. So activation of enzymes and chemical reactions and softening of seed coat. That is the significance of water. Next is oxygen. Why do we need oxygen? To get energy. So same thing, seed is undergoing rapid cell division. This plumule has to form the shoot. The radicle has to form a root. A new plant has to emerge from this tiny seed, and for that it requires energy for that cell division. And who gives it energy? Respiration. And which is the respiratory gas? Oxygen. Coming up next is suitable temperature. Now what significance does a right temperature have in germination? Just now we were talking that germination is catalyzed by enzymes, and enzymes are basically proteins in nature, and they are very temperature specific. Like if you give too much temperature, it's very high temperature, you put the enzyme in that temperature, it would get destroyed due to heat. If temperature is very less, it would get inhibited, it would stop working properly. So high temperature, very high and very low is not good for enzymes, and when it is not good for enzymes, it is not good for germination as well. So we need a suitable temperature, normally between 25 to 35 degrees Celsius. Now ours is a tropical country, so some variation is there between temperate seeds and tropical, but overall a basic range is between 25 to 35. If you give all of this to a seed and its dormancy period is over, then a seed will germinate and give rise to a new plant. Now this germination also is of two kinds. What are the two kinds mentioned here? Epigeal and hypogeal. Now these two terms, don't they remind you of two terms we discussed in the previous class? Epicotyl and hypocotyl. Likewise, germinations are epigeal and hypogeal. But here the story is a bit different. During epigeal germination, hypocotyl elongates. It's opposite here. Remember, during epigeal germination, hypocotyl elongates, and during hypogeal germination, epicotyl elongates. So you have to remember, it's just the opposite. Differences between the two germinations are very important. The second difference is, when you sow a bean seed which is showing epigeal germination in the soil, the cotyledons come above the soil. So you try soaking some bean seeds at home and sow them in the soil. Within a week's time, you will find the cotyledons coming out of the soil. They would also turn green and they would start performing photosynthesis. So this is the main thing which is happening in epigeal germination. The cotyledons emerge above the ground, whereas reverse happens in hypogeal. Try doing it with a maize grain. So a maize grain in the soil and you will find only the plumule emerges, leaves are formed, but cotyledons continue, or the cotyledon rather, continues to remain under the ground. So two differences between epigeal and hypogeal germination. As I told you, it's opposite. In epigeal germination, hypocotyl elongates, whereas in hypogeal, epicotyl elongates. Here cotyledons remain, or they come out above the ground, and here cotyledons remain under the ground. I know it's slightly confusing, so to simplify things, I've drawn a typical seed, its parts here. We did something similar in bean, but I've split it further open to make things clear to you. Now these are the cotyledons. Say we are talking of a typical dicotyledonous seed. This is the point of attachment of cotyledons. Now this is the plumule. The region between the plumule and the point of attachment is epicotyl, and the region between the point of attachment and radicle is hypocotyl. Now we are saying in epigeal germination, hypocotyl elongates. Just take a close look at this diagram and imagine you have sown this seed in the soil and suddenly this area elongates. It becomes longer. Say it becomes this long. What would happen if this area becomes this long? Isn't it going to take these cotyledons above the level? And this is what is happening. Say this was the soil and this area immediately elongates. So what would happen? The cotyledons would emerge above the ground. Whenever hypocotyl will elongate, it will carry these cotyledons above the level of the soil. Reverse happens in hypogeal germination. The epicotyl elongates. The epicotyl is the region between the plumule and the point of attachment. Imagine this area suddenly growing longer. If I sow the seed, if this area becomes this long, it would only take the plumule with it outside and it would leave the cotyledons below the soil. So this is your second point, cotyledons remain below the soil. So next time whenever you are confused, just think of this diagram and picture yourself, imagine yourself hypocotyl elongating, that means cotyledons going above the soil, or epicotyl elongating, that means cotyledons remaining below the ground. Now these are the two main kinds of germinations. In your books you will find another kind of germination, and that is viviparous germination. Now viviparous germination occurs only in some plants like in the mangrove plants. You all know mangroves grow in swamps swamps, marshy areas. So every seed requires air or oxygen for germination, but marshy area, there is no soil air. So if you put a seed in a marshy area, it won't get air or oxygen and it won't be able to germinate. So to overcome this problem, nature has planned a solution for mangrove trees. What happens? The fruit bears the seeds inside, and seeds start germinating when they are still inside the fruit only. Seeds are not sown in the soil, they don't fall in the soil. They are inside the fruit, fruit is still attached to the parent plant and seed starts germinating. When the seed forms a tiny seedling, the seedling becomes heavy, it falls into the marsh. And once it falls into the marsh, it is able to take care of itself. In your junior classes and class 6th and all, you must have studied about breathing roots. How from the roots, breathing roots develop, which are called pneumatophores, which have pores or openings in them. So in marshy areas, once the seedling falls into the marsh, the roots give out breathing roots or aerial roots which come out above and help it in breathing or in respiration. But this was not possible if we put a seed in a marshy area. So viviparous germination is found in mangroves, and it is the process in which seed germinates when it is still inside the fruit, when the fruit is still attached to the parent plant. You study all this from your book. Go through epigeal, hypogeal and viviparous germination. There are also a set of experiments at the end of the lesson which are there to demonstrate whether water is necessary or oxygen or suitable temperature. Go through the experiments. After this, what I've explained to you, you'll find no problem in understanding those experiments as well. So this was all about the lesson seed, the structure and germination. Hope it is clear to all of you. Thank you.